Dear colleagues, on behalf of my co-authors, I present you a report on the following topic: plant secondary metabolites as bioactive substances for innovative biotechnologies. So constant changes in the environmental conditions, they cause changes in the biochemical and the physiological processes of all living organisms, as well as plants. Plants synthesize a variety of substances that have different useful properties and take part in the development of plants. So one of the most important and diverse and numerous groups of bioactive compounds among them is the secondary metabolites, especially phenolic compounds. Natural sources of the pharmacological available metabolites, secondary metabolites, are plants, fungi, and lichens. Phenols are characterized by the presence of the benzene ring consisting of 6 carbon atoms and one or more hydroxyl groups. They, as the pharmacological valuable substances, occupy the prominent place among the secondary metabolites of plants due to their strong antioxidant, antifungal, antibacterial, immunomodulatory, and other properties. Flavonoids and xanthones are heterocyclic polyphenolic compounds synthesized by vascular plants. Non-lichenized fungi and lichens, they synthesize xanthones too. In plants, these compounds, they perform their different functions. For example, the protective role of them to the stress for plants and to the resistance to pathogens. So they participate in the allelopathic interactions and so on. So plant extracts, they are very useful because of these properties for treatment of cancer, Parkinson's disease, the problems of the cardiovascular immune system, and so on. Due to the constant reduction of natural habitats of the plant sources, secondary metabolites are insufficient efficiency and the insufficient efficiency of methods of their cultivation. It is important to find alternative sources of these compounds, which can be, for example, the high reproductive cultures of plant tissue in vitro. According to this, the aim and the task of our research was to study the different species of plants under the aseptic conditions, as well as the use of the nutrient media as the additional environmentally friendly sources of pharmacologically available secondary metabolites. So, according to the information I mentioned before, we use generally accepted methods of the microclonal propagation of plants with modifications to get the plant material. We also use the quantitative analysis of spectrophotometric analysis for the determination of phenols and flavonoids, chromatographic and the spectrophotometric methods for the determination of xanthones by our own modifications. As a plant material, we use the different plants. So the explant of plant of genus Phalaenopsis orchid are used as the source of bioactive compounds. We also took the calamus plants, Acorus calamus species, as the source of plant material to get the compounds we searched for. So the original plants belong to different localities. So they were taken out of nature. Then we got the clones of them. So, and the explants of the clones, explants of these plants were grown into the in their medium, agar nutrient medium, modified for them. So it was the medium Mauritius Kuga. The temperature was 24 degrees and the 16 hours photoperiod. Due to the physiological ability of plants to secrete bioactive substances into the substrates by the root system, as a promising material for the production of phenolic compounds, we used the nutrient medium on which these investigated plants were grown. So, in vitro cultivation of plants was carried out in the research laboratory of the introduced and natural phytodiversity of Permian Botanical Garden, Taras Shevchenko National University of Kiev. According to the information and according to the analysis of the total amount, total concentration of the secondary metabolites, it was shown that there were differences in the quantitative content of phenols between plants of different localities. For example, about the calamus, the first group accumulated the phenols in the leaves and roots. The second one only in leaves. So we can assume that the phenolic compounds were released into the nutrient medium. So we found that the amount of phenols in the nutrient medium on which the Acorus plants were cultivated also increased during the experiment that proved our hypothesis. The total content of the flavonoids decreased over the time in the explants of group 1 of Acorus in leaves and roots. However, in the group 2, there was the opposite situation. So the content of the substances increased in leaves and roots. Studies have not found flavonoids in the nutrient medium in such concentrations that it was possible to quantify them. The analysis revealed that the content of xanthones in vitro plants remained stable or increased during their cultivation. We also noticed the differences in the composition of substances depending on the part of the explant and the plant source. So we found interesting that calamus plants they accumulated only one compound of xanthones. It was the swerhurine. And also, as you can see on the slide, that on the second group of plants, it was the increase in roots and leaves. And in the first group of plants, the increasing concentration of swerhurine was only in leaves. So we can assume that their difference because of their origin of the plants. So the different complex of xanthones was also found in the nutrient media of calamus of different populations. So there was not only the swerhurine, it was also there 3 other compounds. So they differ from depending on the groups. So the content of the compounds decreased during the experimental time. It was found that in the tissue of explants of Phalaenopsis, there were 4 compounds that were accumulated. I mean about the xanthones. So for example, the deglucetine, alpha-mandestin, and others. So in the nutrient medium in which the Phalaenopsis were grown, 2 xanthones were found that were present in the explants. It was the deglucetine and alpha-mandestin. 2 xanthones were also identified, which are present in the trace amount in explant. Also, it was like the swerhurine and the mandestinone A. So mostly they were found in the nutrient medium. The content of this compound in the nutrient medium decreased or was stable with the age of the culture. Therefore, for the biosynthesis in the in vitro plants of the maximum number of phenolic compounds, especially xanthones, it is necessary to regulate the conditions of their cultivation, in the particular the duration of cultivation. So we believe that plants grown in the in vitro can serve as the source of the secondary metabolites, and the nutrient medium can be the additional source that will help preserve valuable medicinal species. Let me not read the conclusions as they were highlighted during the report. Thank you for your attention.